Hello and welcome to this talk about the standardization of flow cytometry used for evaluation of cellular immunotherapy and solid organ transplantation. My name is Mathias Streitz. I'm administrator of the flow cytometry facility in the Institute of Medical Immunology at the Charité University Hospital in Berlin, Germany. And I'm also administrate the immune monitoring via flow cytometry in international clinical trials. But let's come to the topic. Standardization of diagnostic tests and procedures within diagnostic laboratories is the basic principle for the guarantee of highly proved measurements of diagnostic relevant values. That is true for simple staining procedures and also for highly complex analytical processes. But not only in the clinical diagnostics, also in the immune monitoring for studying the effects of therapeutics has become the standardization of analytical methods of high importance. One of our most powerful tools for the immune monitoring has become the flow cytometry. Because it's also one of the most complex methods, standardization is also a challenge. I will show here the standardization of the immune monitoring via flow cytometry for an international multi-center study as an example for the necessary procedures. The one study is a unified approach to evaluating cellular immune therapy and solid organ transplantation. Cell therapy is a novel concept for immunoregulation. In this study aimed at reducing transplant rejection after kidney transplantation. And within the one study, it is planned to use different cell types with immunosuppressive characteristics to promote protologenic responses to renal allograft and assess the biological effect of these regulatory immune cells in comparison to an independent clinical reference group trial. In that trial, it will be used standard medication. The study is performed at transplant centers across Europe and the US. Within the one study trial, it is planned to test naturally occurring regulatory cells, so-called anti-regs, the type 1 regulatory cells, so-called TR1 cells, the tolerogenic macrophages, so-called MREGs, and also tolerogenic dendritic cells, predominantly for their safety. So the second slide here shows the study design. All trials have a runtime of 15 months, whereas a graft rejection is a primary endpoint. Each center is performing the same reference study using a standard medication, including an anti-25 antibody straight after transplantation and medication with steroids, mycophenolate, mofetil, and tacrolimus. At least the complete reference group includes 60 patients monitored over the 50 months after renal transplantation. The cell therapy study is a series of independent trials. Each center is using its own cell product, but the medication protocols are similar. An anti-25 antibody will not be used, and in addition to a reduction of the steroids, it is also planned to reduce the treatment with MMF. Following experimental results for a treatment with immunoregulatory cells can be expected various effects on the composition and function of the recipient's immune cell population. A treatment could result in an increase of recipient regulatory T cell populations, maybe also in an increase of dendritic cell subsets or in reduction of pathogenic memory T and B cells. It could be also increasing signs of operational tolerance, or in a worst case scenario, also an increase of infectious complications. To be able to compare the effectiveness of this alternative cellular therapeutics, the One Study Consortium developed a robust immune monitoring procedure. So we combined different analytical tests, and we performing this test at different time points. At least we have a combined visit prior transplantation and 9 following up visits after the transplantation, where we performing the leukocyte profiling via flow cytometry prior the transplantation and at 8 following time points. We have also defined 3 additional visits around the time point of the cell fusion, and these visits include also 3 different analytical tests as well as the leukocyte profiling via flow cytometry. Within the immune monitoring, we testing the safety. Therefore, we determine the viral load, especially for CMV, EBV, and BKV, and also the level of HLA-DR on monocytes as a sign of immunodepression or paralysis. And we performing immune monitoring to test the efficacy of the cell products. So we combine gene expression determination, the leukocyte profiling via flow cytometry. We determine the occurrence of anti-donor HLA antibodies, or we look at the stability of Tregs via the TSDR demethylation. Within all these tests, flow cytometry, together with different pre-analytical factors, has the highest variability. Therefore, it is crucial to standardize this method. In a paper by Holden Maker, it's named The Model for Harmonizing Flow Cytometry in Clinical Trials. They are described the main issues for a standardization procedure in flow cytometry. The 3 main complexes are the issues of instrument setup, the issues of sample handling, and the issues of data analysis. These 3 complexes bearing a lot of variables that need to be controlled to measure at least accurate and precise data with the least variation among different sites. But we have to keep in mind that a reduction of variability also could lead to an increase of the workflow, and we have to find the balance between both points. In last autumn, there came up a really interesting guide for validation of cell-based fluorescence assays published in Cytometry Part B. And in this paper is pointed out that there's a problem to determine the accuracy of the flow cytometry test, because according to the ISO definition, accuracy is the closeness of agreement between the average value obtained from a large series of test results and an accepted reference value. But the problem is that we have a lack of fully characterized reference material. And at least there's also a problem to find material with variations of target molecule levels. And because flow cytometry is so complex, it is also not sufficient to compare other analytical methods with flow cytometry. At least we can only estimate the accuracy of this test. Within our study, we used the so-called mixed model for the validation and the standardization. It is also described in the paper by Holden Maker. We defined a central immune monitoring lab, that is our lab here in Berlin, and we developed consensual together with the assay performing laboratories and also with our industrial partner Beckman Coulter the panels, and we developed also the SOPs. We did together with Beckman Coulter also the panel validation, and we defined the reference standards. In the second step, in the standardization, we transferred the panels and the SOPs and additional information about the reference standards to the assay performing laboratories. At these laboratories, we did a cytometer setup. there is now doing the sample processing according to the SOP and the data acquisition. After data acquisition, all the data are transferred to the central immune monitoring lab, and we doing here centralize the data analysis, at least only by one analyst, to have a reduced bias via analysis. There's a basic principle for standardization. The basic principle is keep it as simple as possible. And you have to keep it in mind if you answer at the different question that has to be answered. So there are pre-analytical questions. The first question is which matrices we use. At least we decided to doing whole blood surface staining because we are not only interested in the relative values of the different leukocyte populations, we are also interested in the absolute concentration of these cell populations. And we decided to use as an anticoagulants the EDTA because we are doing a surface stain. At least we are using one single SOP within the staining of all panel. It is a simple SOP, only surface staining combined with a leukocyte lysis and fixation, combined with the washing steps to reduce the background. And we had to perform a second SOP because for the B-cell identification of the B-cell population, it is necessary to reduce the serum concentration of the IgM. And we are also interested because there are some rare population to concentrate the B-cells within the sample. Therefore, we added an additional washing step prior to surface staining, but then we followed the same SOP. That enables to use one unique cytometer setting for all the design panels. So now let's have a look on the antibody panel. The first antibody panel allows the enumeration of all major leukocyte subsets, such as granulocytes, monocytes, lymphocytes, and as well NK cells. With the other 5 panels, more detailed analysis can be studied on T-cell activation, T effector cell differentiation, regulatory T-cells, B-cell differentiation, and dendritic cell subsets. In all panels, we are using a unique backbone, CD45 backbone. We need this backbone for the calculation of absolute values, and we are using the white blood cell count of the hematology department as a reference for calculating these values. And in all T-cell panels, we have also a unique backbone by the markers CD3, CD4, and CD8. At least we are using within all the panels for all identical markers with the same close. After fixing the formulation of this panel, the panels never changed, and additional panels will be designed with respect to the backbone and with respect to the used antibody close. All these panels in this study are produced as pre-configured liquid mixes at Immunotech Beckman Coulter and then shipped to the different centers. That enables a high quality for all used antibody panels. Because it is not possible to determine the accuracy, it is necessary to test the imprecision of the panels and the stability. Imprecision tests are the intra-assay tests to estimate a tube-to-tube consistency. There's the inter-operator test to estimate the variance by operator. And there's an inter-assay test to estimate the day-to-day variance. The problem if you are dealing with a whole blood assay, like in this study, is that you cannot use stabilized whole blood for the estimation of the day-to-day variance because if you're interested in markers like CCR7, it is impossible to stain such stabilized blood. Therefore, you have to draw blood each day, and that gives an additional biological variance in this test. This additional biological variance increases the CVs for this test. The stability is tested for the matrix. So you are looking at the aging of your whole blood, and you are interested in the aging of your stained samples. This is a short overview for one of the panels for the interoperator test and the intra-assay test. At least we had really nice low CVs for the intra-assay test. For the most population, less than 10%, and for some populations, less than 20%. Also, the interoperator test is comparable to this intra-assay test. But what does it mean to have a CV up to 10% or 20% or 30%? Is it really good that we have a CV less than 20% for population with variants? For diagnostic procedures, you're dealing with a cutoff value for diagnostic purpose. And for such tests, you have to define the acceptable variance before you're starting a validation procedure. But in the immune monitoring, you do not have such kind of cutoff value. Therefore, you have to report the variability of your panels. So, therefore, we define such precision profiles. At least we plotted the observed CVs for each population in the intra-assay test in the right column here, versus the relative size of the population in the left column here, versus the absolute counted events for each population, and in the middle column to the calculated absolute value. Then we did a best fit, or we looked at the best fit curve and calculated the 95% percentile to find an upper limit for the quantitation. That means for our test, we are finding CV up to 20% if we have a population size about 0.18%. If in decreasing variability up to 5% if we have populations greater than 15.61%. The same is true if you look on the counted event, and that is not necessarily compared to the relative size of a subpopulation. If you are counting 45 events, you will see a CV about 20%, and you have to count more than 1,507 events to get a CV less than 5%. The upper limit shows you that in the worst case scenario, you have to count more than 2,000 events to get a CV less than 5%. This precision profile gives you at least an overview about the imprecision of your test. This graph shows you that beside the interoperator test and intra-assay test, you have an increasing variability for the stability. In the first row in blue, you see the intra-assay test. But with the other colors, in dark red, you see the age of blood 24 hours. In pink here, the age of blood 4 hours. In dark orange, age of stain 24 hours. And in the light orange here, age of stain 4 hours. Let's have a look at a population that is not so great, not so big population. So let's have a look at this CD28 negative CD4 positive effector cell population. We have a slightly increased CV also for the intra-assay test here, but then you see also an increase for the age of stain after 4 hours, but a high increase here after 24 hours after the staining procedure. The same is true also for the age of blood. So you have here small increase of the CV within 4 hours of the preparation after donation of the blood, but a really high increase of the CV after 24 hours. At least we decided then that we have to stain the material 4 hours or within 4 hours after the donation of the blood, and we have to measure all the samples also within 4 hours after the preparation of the sample. After we finishing the validation procedure, we started the standardization, and therefore we had to choose a calibrator. In our study, we are using flow cell beads, synthetic bead particle with a defined MFE value for each channel, but you can use also other synthetical beads like the rainbow beads. It gives you also defined MFE values for all of the fluorescence channels. But we are using this calibrator not only to do a PMT setup, we are using it also for quality control. So we are measuring the flow cell beads each day before the measurement of the samples with the same settings but without the compensation. And we are tracking the results for each channel over the time. If we see then a variance of this MFE value for a channel more than 5%, we are starting again the setup procedure to set up the machine to the target value. After defining the target values, we provided all this information together with the information about the bead lot to the assay performing laboratories. Then we performed the cytometer setup via auto setup procedure at the assay performing laboratories, and then we did an interlaboratory comparison. The results of this comparison are shown here. Therefore, we shipped whole blood samples from the Central Immune Monitoring Lab to all of the assay performing laboratories, and then we performed the stainings to the same point at all these centers. Because we have this large distance between Berlin and San Francisco, we had to deal with 30 hours age whole blood. But with this information in mind, it is really nice to see that we have only a slow increase of CVs for the population with more rare events. So the effector cell population, CD4 effector cell population, but for population they are bigger, like this, we have really small CVs also within the interlaboratory test. All these results, all this procedure is published in this paper and is also nicely to find at this open source platform, Transplantation Research. But after finishing standardization, validation, we are also interested in a panel improvement. Then we are interested to harmonize other studies with our one study. So there's a second huge transplantation trial, so-called BioDRIM, and for that trial we designed also the panels using the one study panels as the basic for improvement. As one example, we used the panel for the T-cell subsets and combined 2 additional markers because in recent publications, it could be shown that a ratio of the V delta 1 and V delta 2 cells give us information about upcoming operational tolerance. Therefore, we added these markers to the one study panel. Then we designed an additional intracellular staining panel, because in the one study we had not a mandatory intracellular panel. We tested once. That we improved by additional markers because we are interested also in the Treg subpopulations. Therefore, we have now in this panel not only a FOXP3 staining and a 25 staining together with CD4, CD3, and 45. We are also staining Helios, granzyme B, and 45RA. All these panels now formulated as multicolor cocktail panels in single tube format with a new dried reagent technology, so-called DuraClub. That enables us, first of all, to reduce the preparation steps, because it's not necessary to pipe up antibody anymore. And we have a higher stability of the samples, because the dried Dura clones are more stable than the premixed liquid panels. And it's now much easier to ship these panels to other laboratories. The one study panels and the BioRim panels are now harmonized in different trials. In the reference study of the one study, we now enrolled almost all patients. So we have now enrolled 60 patients in that study. Then we started now with a therapy study at 2 centers, and we performed a healthy control group. Now applied also other international centers to the one study panels, so that we think that we will have an increasing data pool. In the BioRim panel, we plan to enroll more than 350 patients, and we are using the same panels than the one study and some of the improved panels. And we harmonized also our immune monitoring in smaller trials or in smaller studies in our own laboratory here. So we get at least results from liver transplantation studies, from cardiology studies, or from studies about diabetes and adipositas. But we are also interested in the optimization of the inter-platform standardization. Because we are using bright MFE target, we will have after the standardization procedure at each cytometer the same MFE target for the positive value. The problem is, if you have different filter setups or different laser power in the systems or different laser for the excitation of the fluorochromes, you will see a shifting of the negative population, so that the discrimination of two populations can become complicated. Therefore, we are really pleased that we have a highly discriminating populations or marker for almost the most populations. Here shown for the CD19 positive B cells, and here for the PDCs. Crucial is it at least for that population with modulated marker, especially if there are rare populations like the CD56 high NK cells or the transitional B cells. We did this inter-platform standardization in our own laboratory using liquid antibody mixers and the DuraClone antibody mixers of the same panels. And then we measured the samples on the reference machine for the one study trial, and also at a second platform. On the left column, you see the results for the B cell population and the T cell population, the T cell subpopulations for the liquid antibody mix at our reference machine. In the middle column, for the DuraClone mixes of the same sample, same material. And in the right column, you see the results measured on a different platform with similar settings. The results are highly comparable. To prove our test, we analyzed a healthy cohort. In that cohort, this time we included 52 patients, and these 52 healthy control individuals, and that covers 26 males, 26 females, and we can split up this in 5 different age groups. So we have a well gender and age matched group. Now we did a one-way ANOVA to identify the 10 populations with the highest discriminating capacity between different groups. And you see here the 5 different age groups. So in white, the group from 20 to 30 years, and in black, the group from 60 to 75 years. And if you look on the left side, the heat map here, we are starting on the right side with the younger samples and have on the left side the samples of the older population. And for age markers here, we see an increase, because it turns to blue, of that subpopulations within and a decrease of this population within an increase of the age. Only for 2 of these populations here is shown that this population increasing with age. The founded result fits very well with the published results in the recent literature, because it is not surprising that naive Tregs decreased with age, whereas the memory compartment of the Tregs is increasing with age. We did also an ANOVA, one-way ANOVA within age-corrected samples, or with the age-corrected values, and therefore we found also interesting discriminations between the female and the male groups. At least in that sample is shown that in the male group, you will find more effector cells, where the population with effector cells are decreased more in the female phenotype. Not only to prove our test, it is necessary to use that healthy control group. It is also necessary to further compare our patient cohort with that age and gender matched controls, because you see the differences in certain populations by aging. As a second test, we used the results of the pre-transplantation time point to look at the subpopulations also with the highest discriminative capacity. So here on pink, you have the healthy group, and you have the kidney transplantation group in blue, and the patient with a non-confirmed rejection in yellow. In that patient with a time point prior transplantation, you find a decrease of almost 7 main population, including also the PDCs, and you found an increase for 2 of this population here. Comparing the results with the healthy control, you can clearly see for the PDCs that you have an increasing of the PDCs while aging for the healthy controls here in black, and you have a much more decreased level of PDCs for the patients before transplantation. Using that 7 subsets, we performed a principal component analysis to identify a phenotype for these different groups. Surprisingly, we found that the healthy phenotype is more comparable to the yellow phenotype here of the kidney transplantation group with a non-confirmed rejection, whereas the phenotype of the patients that didn't got a rejection prior transplantation is much more different from the healthy control and from the patients that got a rejection. But these are only the first results of the study, more to prove our immune monitoring, because most of these results are published and we can compare it. So we are now highly interested in more to combine the data analysis using the data of all analytical tests, for instance, the TLDR demethylation or donor antibodies or the other analytical tests. And we are also working on the further optimization of the test, because we are highly interested in reducing the variability and improve the standardization procedure and also improving the analytical depth of the data analysis. We are interested in the further development of other panels using the one backbone, and also in the optimization of the standardization, maybe via other reagent platforms. We also hope that we can help to develop a fast and reproducible whole blood functional assay test in pre-coated dry down tubes incorporating stimulus. And we will go further, or we'll move from the peripheral blood test also to graft and biopsy staining with our panels. And as a really big complex of interest, we also look at the post-analytical issues, because we are highly interested in reduce the bias while data analysis. And we hope that there's a development of bioinformatic tools for data interpretation. There are 4 main complexes. The main complex for reducing bias while data analysis is the automated flow gating. So there are 2 possibilities, a supervised and unsupervised automated flow gating. But it is also necessary to develop other statistical tools and classification algorithms and improve the data visualization. At the end, we hope that, especially with the harmonization within other studies, we can doing a data sharing of this increasing data pools using the back panels. At least I shouldn't forget to point out the most important factor for the variability within the test. This is the communication and the training. And I'm really happy that in our consortium, in the one study, we have a really good communication between all partners. We have a really well ongoing project, and we have a really great collaboration between the assay performing laboratories, our industrial partner, and the central immune monitoring. So I'd like to say thank you for your attention, and I would move forward to upcoming questions. So the first question by James is here. Where were samples collected? Did you control for logistical variables? Yes, in the centers we are doing, because we are doing an on-site staining of the samples, we have really short ways from the place where the sample where the blood is drawing to the staining place. And for the inter-laboratory test, we controlled also the logistical variables, if possible. So we reported the change of temperature within all these transport procedures. So if there are no additional questions to this talk, I would say thank you again. Yeah, and I wish you a good day.